Good afternoon, first of all. Thank you for coming today. And my project has been on engineering self-assembling peptide materials, and I'll explain what those are in a second, using simulations and using diphenylamine as kind of like a case study for that. And I've been working with my mentor, Ji-Hoon Jen, and Professors Scott Shell and Patrick Doherty in the Department of Chemical Engineering on this project. So, as promised, what is a self-assembling peptide material? So the peptide part of it comes from those biomolecules. As Erica had said, nanostructured materials are pretty important nowadays, and they have a lot of potential applications in both energy and biosensors. But there's a lot of questions about the health implications of these nanomaterials. And so one way that people have looked at circumventing this is using biomolecules as their inspiration. And because these are like using the building blocks that are found in biomolecules as inspiration, because these building blocks are found in our bodies, there's a lot less potential for them to be carcinogens and stuff like that. And so self-assembling peptide materials, the self-assembling part simply means that they come together without much outside force involved. You don't have to put energy in to make them form. And so they found that peptide materials can form into a wide variety of structures, including nanotubes, nanofibers, nanotube forests, nanospheres, nanovesicles, etc. As I mentioned, they're biocompatible, they're relatively inexpensive, and they can form a variety of structures. And so our target molecule, and the one that we've been looking at, is diphenylamine, and it's a really small peptide. Its molecular structure is shown right here. And it forms into nanotubes. And we did a lot of work about a year ago on this and used simulations to study it and figure out kind of in its earliest stages what drives it to form into these nanotubes and how does it oligomerize. And what we found is that diphenylamine is a pretty unique molecule in that it has a lot of properties in one small little peptide. So right in here it has a hydrophilic region, so that means it likes water, along the backbone, and then it has these very hydrophobic side chains, meaning it really dislikes water on this side. And we found that it was a combination of these properties working together that caused this peptide to self-assemble, at least in its earlier stages, to form ordered structures. And so, based on this, we move forward and ask the question, what happens when we start tweaking the hydrophobicity of that side chain? And so the easiest way to decrease the hydrophobicity a little bit is to add an OH group on the side of this peptide, right here or right here, and that makes it a little bit less hydrophobic. And what we wanted to see, and one of the questions we wanted to ask is, does it make a difference if we put this OH group on one side of the peptide over the other? Because our previous work had suggested that the two side chains, while they're exactly the same in this molecule, kind of have different interactions that influence the behavior of its self-assembly. And so we do this first using simulations. So we did 2 simulations on this variant system, one with 2 peptides in a box of water just floating around, and one with 12 peptides in a box of water. And we did that on the 2 order level to see what happens with just 2 of these, and then the 12 peptide level to assess does higher order structure play a role in its assembly, and are there any differences between how it interacts when there's 2 or 12 peptides around. And we can use several analysis techniques to look at this system. The first of these is a potential mean force curve, and this just gives us some statistical quantitative information about how much these peptides really want to come together. Do they want to stay far apart? Do they want to aggregate, etc.? And then we can use another statistical analysis called clustering to look at what are the most frequent structures observed. So when 2 peptides come together, what sort of structures do we see? When 6 peptides come together, what's the most commonly observed structure? And then finally, we have visualization. And I think, yeah, since I had to transfer this over, this video doesn't work, but we can visualize the peptides in the box. We can take the water out when we're looking at it, at least, so look at the peptides clearly and see what happens when they're coming together and apart. And so the first technique that I mentioned, the potential mean force, gives us some information about whether or not these peptides want to come together. And what we find, and so let me describe these graphs to you real quick. So this is just an energy landscape. And you can think of this as lowest energy is here, and if you You can think of molecules as a really lazy person. They want to be, once they get to the lowest point, they don't want to leave because they're going to have to go up a hill. And the steeper the hill they have to go up, the less they're going to want to leave that spot. So if you see a really deep minimum on one of these curves, that means it's really going to prefer staying there because it doesn't want to waste the energy to get out of that minimum. And what we see here is that compared to the original, FF is the original peptide, and Y just refers to whichever side chain has been changed. And what we see is that these are pretty similar. We don't see a big difference in the way that these minimum look. And so we can conclude that they have the same aggregation propensity, at least to come together. However, when we started to look at the actual structures that these peptides formed, we found a slightly different story. So while they still wanted to come together and aggregate, they actually were slightly less ordered, especially when they started to come together in groups of 5 or more. So on the lowest level, the structures came together in a very similar way to the original diphenylamine peptide. But here you can see they're just kind of, well, there are some, these green parts are hydrogen bonds. They are rather disordered, whereas in the original diphenylamine peptide, we saw them stacking into ladders with the backbone, this orange part on one side, and the side chains, this purple part on the other side. But here they are rather disordered. And so the next step was to do some experiments and see if we could see this behavior reflected in the way that these behaved in real life and experimentally. So we start off with the peptide in a powder form. We add a solvent to it to dissolve the peptide, and then we add water on top of that. And this water is what, at least in the case of diphenylamine, causes it to self-assemble into nanotube structures. And so in the case of the variants, we want to see if these nanotubes form or if any nanostructures form. And so as a control, we did the original diphenylamine peptide. And you can see up here, this is dissolving it, as I said, in the original solvent. You can see it's pretty clear. It dissolved rather nicely. And once we add water to it and let it age for a couple days, it might be a little bit difficult to see, but there's these little spindly things. And those are actually crystals that are forming in the diphenylamine nanotubes. And that's what they reported in their original experiments. However, rather unfortunately, the YF, which is the peptide that we were able to order to do experiments on, does not dissolve in the original solvent. And when we add water, it forms these kind of like logs in solution, which we suspect are some kind of surfactant like thing. We think they form micelles in water. And there's other papers that verify that if you just put this peptide in water, it forms micelles. However, the next step that we would want to do here is to find a different solvent that will actually dissolve the YF peptide quite well, so we can get something like this to start off with, and see if we can, after dissolving it, if we can induce self-assembly into crystalline structures by adding water. And so, in conclusion, we find that at the smaller oligomer level, we see rather ordered structures. Here's one of the former structures, and as I was saying, you can kind of see that they're lining up rather nicely with the backbones right along here, or the orange parts lining up on one side and the purple parts lining up on the other side. But as soon as they start to get to higher orders, that hydrophilic side chain causes a lot of disorder, and that kind of prevents them from forming into these more ordered crystalline structures. And we predict that that influences their ability to form nanostructures. So I would like to thank my mentor, Ji-Hoon Jun, for helping me for the past 2 years in my simulation lab with Professor Scott Schell. I'd like to thank Professor Schell, he's not here today, but, and Professor Doherty for their help and support and mentorship over the past 2 years. A special thanks to Sarah Elliott for giving me, she's right here, she's also right there, helping me out with all of my experiments in the lab. I know it may not be easy putting a theorist into a lab and helping them set up, but she was very patient and very helpful. Erica for all of her support over the past, not just 2, but 3 years. And the Arnold and Mabel Beckman Foundation for the funding that made this project possible.